Fine.
All right, we are now recording, and thank you very much, Sam, for agreeing to give a Build a Sim seminar. And go ahead.
Okay, thanks. So I will give a little bit of introduction of myself here. My name is Sam Weiss Evans. I'm a senior research fellow at Harvard University's Program on Science, Technology, and Society. And I've been involved in the synthetic biology, bioengineering area for over a decade now, looking at the ways that areas of synthetic biology become matters of concern and how those are tied to particular ways to govern those concerns. My work largely focuses on security issues, but I have a strong line of work in the non-security areas of synthetic biology as well. I'm going to try to keep this presentation to about 30 minutes, which means I'm going to start my timer here. But I encourage you all to interrupt me along the way, and I look forward to the conversation afterwards, because the whole point of this talk today is to try and think about how we change the way that we govern synthetic biology and the spaces where synthetic biology enters into other types of governance systems. So thanks for having me, and yeah, let's get started. So I called the talk Ways to Become Involved in Experiments in Synthetic Biology Governance. And I think to start, it might be helpful for us to think about why we want to even experiment in governance. Like, don't we just have a governance system in place already, and we kind of build from there? Well, yes and no. So we have a lot of ways of thinking about what needs to be governed in synthetic biology, but many of those systems that we have in place, from biosafety committees to conflict of interest policies and others, are based on a set of outdated assumptions, or what I would consider largely outdated assumptions about the relationship between science and society. The biggest of these is the idea that science is somehow done separate from society, that you have the kind of stuff that goes on in the lab and the work of scientists and researchers, and then there's the kind of move to the outside world. And that simple kind of starting place has a whole bunch of ramifications for how we think about science need. We might want to be thinking about the need for experimenting in governance is that there are often really big policies on policy changes that come about, and those are often they often come about because of big events, 9/11 or anthrax attacks or other kind of pivotal moments that then instigate radical policy change. But those changes are often done very quickly and without a lot of time for thought about ways to do them well. And so an experimental approach is a way of kind of expanding that process of trying out new ways of governing that allows us to both be at smaller scale, trying things out, and more systematic in the process that we do this kind of redesign of our governance systems. And then finally, that drawing out and slowing down allows us to, and being more systematic, allows us to look at our governing system as an experimental space. And by that, I mean focusing on documenting and learning, on sharing what is and isn't working, and on iterating these experimental projects. So if you want to do an experiment, what do you need? Well, this is a paper that came out in Science a couple of years ago, with I and a couple of many other biosafety, biosecurity people from around the world, thinking about the importance and need for experimenting, particularly in biosecurity. And we talk about there being 4 aspects of experimenting to focus on. One is starting with outlining the assumptions that you're making about science and society in your experiment. So What is it that you think are the assumptions being made currently? What needs to change? What are the alternative assumptions? And then have a systematic process of documenting how the experiment is going, what's working, what isn't. That documentation then allows you at various points in the experiment to step back, reflect on it, and then share with others what is and isn't working in this experiment. And that then allows you and everyone else to iterate upon the design of them as they go forward. So this is all kind of high level stuff. And what I'd like to do is get down a little more into the details. I'm going to take one assumption that we make a lot in synthetic biology and bioengineering, and actually much more broadly in most of science governance today. And that is the focus on a lot of these governance systems on the intent of the researcher and the intent of the research project. Now I'm going to show you this one, but throughout my talk, what I would like you to do is to be thinking about your own experiments that you might like to do in synthetic biology, bioengineering, governance. And at the end, I want to hear about those, because I would really love to see whether and in what ways you all find this talk generative for your own work. Okay, so just get started. What I'd like to do is, I don't know if I can easily see the share of hands here, but I've got a question for you all. So what do you think is more likely? That someone purposefully creates and uses a bioweapon, or that unintended consequences of bioengineering end up re-inscribing the inequalities that we already have in society? So if we can maybe try raise of hand here, if you think that someone purposefully creates and uses a bioweapon is more likely, raise your hand. All right. Okay. And then if you think that unintended consequences of bioengineering are more likely to reinscribe inequalities, raise your hand. Not surprised. Well, I am surprised though. Not and surprised. Why is that? Why is that? That's because our governance system, especially around security concerns or ways that our technology might harm, is really heavily focused on the someone creates a bioweapon. And it's barely focused at all on the unintended consequences question. And so this is a big issue for us that I don't think gets enough attention, because the idea is, because we think that we are trying to do good, therefore we are doing good. And that creates a set of blinders in the whole research process on the ways that we might think otherwise. So two main myths about intent that I want to be unpacking throughout today. First, the idea that researchers only need to be worried about their own intent, not those who might use their knowledge or technology. So if the researchers are trying to do good, then that's good enough, and it's everybody else's problem about what to do about the knowledge or products that are generated. And myth two builds on that, and that it's from the perspective of the researcher. If the researcher believes that their intent is good, then by default, the knowledge and the technology that they produce are good. So there's this coupling of good intent means good knowledge. And that undergirds a lot of our inattention to the ways that good intentions might cause harm. And so what I want to do is I want to decouple this idea that good intent equals good content, and figure out what we might do differently. And to figure out how to do that differently, we might focus on any one or many of the different stages of the research life cycle. So we've got, for example, idea generation. most of this work on idea generation for new research projects is in the form of like how can we make the world better, um, and and how can we then make the world better in a way that somebody might pay us to do it, and that often leads to really proselytizing the potential goodness of the research that could be done, rather than starting from the beginning, thinking about the ways that it um, might not be such a good idea. Similarly, in funding, while there is basic research funding for broader aspects of research as part of all National Science Foundation grants, for example, those rarely are systematically developed with social scientists and others who have a deep and broad knowledge of the relationship between the social and political and scientific. And if they and what attention is dedicated to those broader aspects and funding programs are often kind of the downstream effect. So it's, again, reinscribing this idea that science is separate from society, that basic research is stuff that's done in the lab, and then we can focus on the all that other stuff downstream after we do our research. So that then logically, if that's the way a lot of funding programs are structured, it makes sense that the conduct of the research is itself also largely focused on the scientific, on the lab, and not on the social contexts within which that research is done. If you think about oversight, the idea of intent being all over oversight issues comes into play if we think about in biosecurity, one area of oversight is around dual use research of concern, or the potential pathogenic care and oversight policies. And these, again, are based on this idea that, well, the researchers are trying to do good, but if they're working on these specific types of organisms or doing these specific types of experiments, then we need to have particular oversight of them to ensure that even though they're trying to do good, that they might cause potential harm. But it's only focused on that narrow set of research. We can also focus on other areas of oversight, like in the United States right now, a lot of work is on a lot of attention is being put at the oversight stage on foreign nationals. And there's this idea that your nationality is tied to intent. So if you're a foreign national, you might be more likely to want to use research in a malicious or nefarious way. And so there are all kinds of policies being rolled out to have a deeper look into the research process from this idea that intent is somehow tied to nationality. Again, publication and production rarely focuses on the detrimental sides of doing a piece of research. The results are usually about the positive aspects of it. And then training, I put last. I could put it first, I could put it in the middle, but that's the point. Like, training is around this entire research cycle, again, is largely based on this idea of you're doing this research because you want to improve the world in some way. And it's taken as a given that that's, in a way, what we're trying to do, rather than starting from an assumption that says, that says one person's good is another person's bad. That understanding that intent of one group might have the opposite effect on another group. And so we have these governance systems in place, ideally, to manage the tension between this idea that what you might think is a good, someone else might think is a harm. And then how do we design our governance system in order to manage that tension and not deny it and not eliminate it by just saying, of course, everybody is trying to do good here. Okay, so then that leads us to a question of, okay, so if I'm concerned about the way that intent seems to be structuring a lot of the ways that we govern synthetic biology today, then what would change if we made a different assumption? If we assumed that harm would obviously come from research as well as potential good. If there was always this idea that and what constitutes a harm and what constitutes a good will be contested. If I make that assumption, what should change? And through that, what experiments can we do to test out how those changes might occur? Okay, so for the rest of this talk, I'm going to go through some examples of the different stages of the research life cycle. Oh no, I should say, I'm not really paying attention to the chat right now, but if people have questions that they want me to, I'll take a second here to address. I'm happy to do that during the course of the talk too. Okay, I'm not going to address those right now, but do speak up if you want to. Okay, so so for the rest of this talk, I'm gonna give a couple of examples of experiments that I have been involved with in some way or another around this question of intent at different stages of the research life cycle. And I'm going to break these examples into two columns. One is about how we can make those changes happen, and the other is about how can we be reflexive about the changes that we suggest. So in other words, one is, so if we think of the whole project here as, I think we're making some assumptions about the relationship between science and society that don't really hold up anymore. I want to change those assumptions. Here are some new assumptions that we might make instead. Let's try these out and see what happens. And then the reflexive bit is saying, am I doing more harm than good myself in trying out these different projects? How do I know that? And whose voices should I be listening to in that process? Okay, so let's think first on the idea stage. All right, so idea generation. I was involved in a project, I'm not really much anymore, with Kevin Esvelt, but this was back in the very early days of the development of synthetic gene drives. And this is back, 2014, 2015. We were trying to think of, well, if we're concerned about the potential development of gene drives, for anybody who doesn't know, I think everybody probably in this room does, but gene drives are a way to propagate a specific genetic mutation or change throughout an entire wild population through biasing inheritance. So Kevin had this idea of creating the synthetic gene drive, and then very quickly thought this might not be such a great idea. How do we then think about how to develop the research of it in a way that is attuned to the contesting concerns about whether it's a good idea or not? So a lot of this happened through conversation, the idea generation about how to move forward with gene drive research in that group revolved around, well, what kind of body could we work with that would push back against the kind of overly scientific framing, that would be attuned to all of the complexity of the way a gene drive might work in the world, and that had the power and authority to speak up against us. And we settled on the idea of enrolling, or at least talking with, the communities of Martha's Vineyard and Nantucket, small islands off the coast of Massachusetts that are inhabited by a small set of local residents and a large set of very wealthy people who have kind of second homes on the islands. And the idea was that these groups are often powerful people in their own right in society, and if they speak up against gene drives, that would be a voice that would need to be listened and heard to. So just seemed like a good idea to kind of move away from this idea that just because we are trying to do good, we are doing good. Um, but it's a small project. It's a single case. There is a ton of funding for gene drive research right now, and it's being done by many other projects around the world. So there's a question on, like, well, what is it that we're doing in this project that could be like, it is by focusing on the small project, in other words, missing the bigger conversations that we need to be having about the ways that we should be governing gene drives. If we look about funding, so I sit on the advisory committee, for the DARPA program that is just wrapped up on safe genes. So this involves gene drives, but other ways of making gene editing safer by design. And I agreed to be on this advisory committee in part because I was really curious in the ways that DARPA is trying to make itself as a funder of research more attuned to the ways that social and political aspects of the research that they fund are attended to. Like, how does a military funder make space for people to question, is this a good idea? What ways are we complicating and unnecessarily maybe directing the research down lines that may not cause military harm, but may cause other types of harm? And I'm happy to talk more about that if people want in the Q&A. But the reflexive part here that I was concerned about, and I will always be concerned about with this kind of work, is by being part of this this advisory group, and by working with this with military funding, am I just continuing to enable the militarization of biology? How do I ensure that the work that I'm doing isn't just a process of giving credibility to research that I might otherwise contest or want to broaden out and expand other ways? If we think about the oversight moment, so I talked about dual use research concern policies before, but okay, so if we're going to expand beyond the sets of experiments of concern and agents of concern, then is it more policies that we need to think about? Well, all research being dual use. I didn't put this up, but I've got a piece coming out in the next, or the current issues in Science and Technology, called, what's it called? When All Research is Dual Use. You can see it. You can see it out in a week or two. And there I talk about how the new National Security Commission on Emerging Biotechnology has this opportunity to really rethink some of these really fundamental assumptions that we're making about the relationship between science, security, and society. And that the commission itself has, by being under the president, has the authority to really give a lot of weight and credibility to the need to systematically attend to and rethink these assumptions. But the concern in making this move by writing this paper and trying to get the attention of the commission, one of the concerns that I have is that I don't want to create more top-down hierarchy, basically, about requirements for you to satisfy in order to get more money and things like that. What I want to do instead is I want to show and open up the idea that what we need is not top-down policy, but we need top-down cover for the people who want to be experimenting in other ways of thinking about potential security concerns to try those experiments. And if they don't work out, like, for that to be okay. In other words, for it not to be a, like, if you don't get this right, then everybody's going to die kind of mentality. Because that, while that might be true in some certain areas, that's not probably true in the kind of broad unintended consequences area. It's much more likely that um, by 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 testing out things, at the very least, we might we might just reinscribe the existing inequalities, right? Which is probably what's going to happen anyway. Okay, so if I move on to, just got two more here, and then I want to turn it over to you all. Publication and production. So I lump these together because I think that it's interesting to think about things like the production of journal articles and papers, along with the production of the products that synthetic biology companies are making right now. So one active conversation in the security world moment is around companies that are producing genetic sequences for hire, basically, and the question of how do you screen those sequences to understand what people are buying, why they're buying them, and how they might be sequences of concern, even if they're not tied to the known kind of lists of agents of concern. And again, there is a questioning here of it's not necessarily that that rather than assuming that intent is the that if they're a legitimate group, then they must be doing good work with it. There's a kind of added layer in the screening process to say, well, but might our screening system itself be, might we have built in capacities for the system to be easily misused if somebody wanted it to? And so working with a group at the Engineering Biology Research Consortium on this effort, this was an attempt to do a red teaming exercise of these gene synthesis companies that then never actually went forward. And we're working on a paper about why that was at the moment. But that's one example of the publication and production process. This yeah, let me go through the other two, and we can come back to these if people want to hear more. The second one, so there was a paper out a while ago on purposefully creating mutations to SARS-CoV-2, and it was a protocols paper that really laid out the steps that you would have to do this in some detail, purposefully to share the process. And there was very little attention in that paper to the potential safety and security concerns that might come from widely disseminating this process. And so a group of us got together to write a paper contesting that. And the experiment here, in a way, is to have a public rebuttal to the ways that journals oversee the potential security concerns in the papers that they publish, and to expand that oversight of potential concern, again, beyond known lists of agents of concern, to consider more widely these questions of process development, like in that paper. The third one, I'm not going to go into much detail here, but another paper where we were trying to work with risk assessors. There were some social scientists working with risk assessors to try and think through in what ways should we be redesigning the process of environmental and ecological risk assessment to adapt it for potential field trials of gene-drive organisms. And there was a lot of debate around what constitutes an adequate risk assessment, and a lot of that was around, well, whose framing of the reasons why we want to have gene drives in organisms out in the field should get included in the risk assessment. So that then directly leads to, well, what counts as a risk, and what counts as an acceptable risk. And we were arguing that at that problem formulation stage, that critical first stage of risk assessment, there needs to be a lot more attention and systematically design the process of problem formation to be attending to these multiple problem formations. But all of these, the reflexive bit here, all of these that are concerning for me that reflexively is how we are by showing the ways that this work could be potentially used for harm, are we ourselves creating information hazards that could then be exploited by others? And so we're as we're doing each of these projects, we're also reflecting on, well, how much do we actually talk about this or not? And that conversation, I think, is a very needed conversation, but again, one that we're not often having in a lot of our research. I feel like I've talked too long, so I'll make this very brief. But in the training side, to wrap it all up, I think that there is so much that we need to do to systematically redesign the training of our scientists to be more reflexive about the ways that the ways that their research are what they research is already set through a lot of social, political, economic practices and organizational contexts, and that the knowledge that they generate has political and social power in the world, and that knowledge needs to be we need to think carefully about what knowledge we want to be bringing into the world and how that might reinscribe political, social inequalities, but have other kind of consequences that might be entirely, while unintended by the researchers, might be entirely anticipated by people who have been systematically underrepresented and made vulnerable by past research efforts. So my work on a graduate seminar I teach called Beyond Don't Be Evil, attunes engineers and PhD scientists to this process. And then the work in iGEM that I've been involved with for a long time on the human practices side, as well as safety and security, is again this experimental space every year to iterate on how do we get the teams thinking through incorporating the broader aspects of their research from the initial design of their projects through to the realization of them. The reflexive side here is, how do we know when we've gotten too much attention to the ethical or the scientific or the social aspects of the training? I still think that there's too much emphasis on the scientific, but that's just me. But how do we make that balance is a question that I think isn't being asked enough. So we don't have good answers to it. So now I want to hear from you. What governance experiments would you all like to run? What thoughts do you have about the ones that I'm involved with? And yeah, let's have a good conversation here. Thank you very much.
Thank you so much, Sam. There is a lot of questions in chat. If you could read a question before you answer it, that's going to help people watching the recording.
So you want me to read them out, or do you have ones you've particularly noticed?
I'm looking through. Some are just comments, but there's some questions, so maybe I can read some for you. So yeah, I see the votes were mostly for the unintended consequences, just like the show of hands. Yeah. I see a lot of people voting for unintended. Okay. First question: is it possible to produce bad knowledge?
Well, I would say simply that knowledge is always contextual, and so I think yes, it is possible to produce bad knowledge, but in part that's because it depends on from what context you're viewing the knowledge is good or bad, and I think that if you are I'll give you a good example. So I should always have this clip ready to show everybody. But there was a meeting in the Mice Against Ticks project on Nantucket, a town hall meeting, and Kevin and another researcher were up front talking about um, how to design a potential gene drive, and whether it was okay for the drive to have trans genes in it. So whether it's okay for you to take genes from one species and put it into the mouse to make, the idea is make the mouse immune to Lyme disease, so that the mouse is no longer a vector for ticks that want to that carry Lyme to humans. And Kevin does a straw poll of whether people in the room would prefer for the mouse to be to have to be focused on just focused on Lyme, or is it focused on trying to make it immune to all ticks? So regardless of the disease, no ticks would like to bite it. And then nobody raises their hand to say, and then he asks, should we just not go forward with the project at all? And nobody raises their hands. And he's like, really? I thought there might be some contention to that. And then somebody in the audience gets up with the microphone and says, if you are in this room and you find that nobody can object to a statement like that, then you do not have the right people in the room. And I thought that was a really, really important moment in this research project, because it's showing you that if everybody thinks that what you're doing is good, then you don't have the right people in the room. It's only once you get people saying, I don't know if this is a good idea, that you really know that you're onto something. Because what you have to do then is you have to figure out not how to convince those people that it is a good idea, but how to manage that they might be right, and how to design your research process such that if they are right, the potential harms would be minimized.
That is a very good point. We see sometimes in our community too. The next question kind of builds on this one. I second the bad knowledge question. Is there ever bad knowledge, or is it just bad use?
Yeah. Well, I mean, I think I kind of answered that in a way.
Yeah.
I will, as we're going through these questions, well, I really want to get people to raise their hands and join in and talk to me too, because I'm happy to read text, but I want to interact with you in person. I don't know what the mano a mano is in Spanish for person to person.
I second that. It would be fantastic if we had more mouth conversations, but we can move through text too. Next question is fantastic point about intent versus nationality. Do you know if this is our unique US perspective, or other countries have similar bias in analysis?
Well, I know that we've got people from the Netherlands here, so I'd be interested in their perspective on this. I am largely based in the US in my work. I would be interested to hear from others. My suspicion is that there is, in general, a rise in attention towards nationalism in quite a few countries right now. And while in general, I think I would push back on the kind of being a national necessarily means that you might be more concerning, I would push back on that in two ways that probably a lot of people aren't. So one is remembering that a lot of the potential harm might come from your own people, and second is understanding that it's a variation. So often the case is like science is global, therefore science doesn't adhere to national boundaries. And I kind of get that, but at the same time, I do think that there are more nationalized ways of producing knowledge, particularly when it comes to regulatory science, or when it comes to particular science that is used as advice within policymaking in one form or another. And I think that um, that coupling of the, well, I guess, am I counteracting myself here or contradicting myself? The intent of the production of knowledge is often tied to a national perspective, is one way that I could say it. But yeah, let me see here.
That I like that. I like that answer.
Mm-hmm.
The next question, how should synthetic cell community proceed here? Oh, I like that one. I want to hear your reply. So the rest of the question is, there are dual use concerns, but we are so early that not much is publicly discussed. Should we talk safety and security in public, or should we keep those conversations internal as long as possible?
Right. Yeah. So we can take a lesson here from the from other types of conversations that have been happening in the genetic engineering, bioengineering community for a very long time. And that is, don't, my argument would generally be, don't try to assume that you've got all the expertise to figure out these questions yourself. Like, assume that the expertise for trying to figure this stuff out resides in a much more diverse community. And therefore, you have to engage with that community in conversation to think about what are the potential ways that we might need to be concerned about the research that we're doing. And again, I think if we look at this kind of most likely scenario is that unintended consequences are going to be where the problems lie. If you've in that framing, you always, like, unintended consequence, it always needs a subject, who is in doing the intention, that means that the consequence is something that they didn't foresee. Because if we put that into context, then we can immediately see, well, it was unintended for this group, but it was entirely anticipated by another group. And so unintended doesn't mean that it's unforeseeable, right? It just means that it's not foreseeable by the group of people that you've got in the room. And so the more diverse perspectives that you have in the room, the more likely it is that you're able to foresee potential consequences, and you can no longer claim that they are unintended, because you can still claim that they're unintended, but that doesn't absolve you from the responsibility to do something about them, which is the way that unintended consequences work right now.
That's a great point. The next question is, could you please speculate on how would we even approach dual use concern analysis for a field like ours? Synthetic cells are chassis orthogonal to natural cells. So how do we even go about analyzing possible threats?
Right. I mean, that is exactly the right place to start. So if we think about, like, the ways that security concerns have been structured in biology over the last 50 years, there have been specific moments where we have had to make a policy choice on the ways that we should become concerned about biology. The first one in the 1970s, we made the decision to say, oh, look, there's like so this is, again, for situating people's memory, if you're that old, the 1970s, we were negotiating the Biological Weapons Convention, but it was also, the moment of Asilomar and this hand-wringing of the genetic engineering community on how to self-govern themselves. And the Asilomar meeting, where a bunch of these genetic engineers got together with a few lawyers and a reporter or two, was a process for them to say, if we don't get our act together, then the government's going to impose lots of stringent oversight on us. So we should figure out how to govern ourselves. And they made some really critical decisions that have reverberated to this day. They said, look, we're not going to focus on the security issues because yes, that's very, very important. We need to be worried about that, but that's not what we're doing. We're focused on basic research. And so they totally threw out the window all attention to potential security concerns. And then they said, what we're doing is just stuff in the lab. So all we need to be worried about is not all of the broad social and political and economic stuff. It's just because we're just doing research. So we throw out all of the social and all of the ethical and stuff. And then they say, okay, since we're just working on the lab, then really it's just making sure that our researchers are safe, right? That they don't get needle pricks, that there's appropriate containment for whatever level organism that we're working on. And so, okay, so what do we need to do that? We need biosafety levels in our laboratory systems. We need a biosafety committee. And okay, we'll give some kind of credit to the public, and we'll let you have a member of the public on the biosafety committee, but we don't really expect them to do anything, right? Obviously, because they don't have the technical knowledge. So it's a fig leaf to public engagement. That's the way it works today. Like not a whole lot has changed. And this is what I'm talking about the need to question these assumptions, because we specifically designed our system to not attend to the security concerns, to not attend to the social and the ethical and the political concerns. And that is deeply embedded within the way that we are trained and the way that we design and conduct our research. So in answer to the question, like, how do we go about starting to think about like concerns in a field like ours? I would say you start the conversation. You sit down, you start thinking, well, what are some assumptions that we're making here? So we're saying that, we are working on an orthogonal axis, and we think that it's important to work on an orthogonal axis. Why? Is it because that provides innate security? Well, how do we know that it provides innate security? And more importantly, how do we know if that isn't, our safety? How would we know if that is not actually the case? So how do we know if the assumption that we're making is actually wrong? And how do we design purposeful monitoring systems to put in place that make the opposite assumption and say, well, if what we're making, if we make the opposite assumption, then this oversight should produce a red flag for us. Yeah.
So I guess the next question kind of relates to how should we do it. The next question is there, or could we try to engineer an international safety framework, something beyond the country rules for the whole synthetic cell community? We're already working together.
Yeah, so I mean, I think that this is happening in some forms or another, but I would, in going up to the international level, one of the things that I'm trying to argue for in a lot of my work right now is not necessarily the creation of these kinds of frameworks, but what it looks like to create a framework that then can be modified and iterated upon as it's implemented. So in other words, there's often a lot of attention into creating like what is the statement of our culture, or what is the framework for assessing a potential safety or security issue, or like what's the ethical framework we're going to use in this process. And just like the first or the second Iraq invasion, like there's lots of emphasis put on like how do we go in and win, and not a whole lot of emphasis put on like, okay, so after we've done that, like how do we actually maintain the system, and how do we build it up again into thinking about all of the deeper changes that need to be made to continuously and seriously consider things that go outside for instance, the list-based process that we have of identifying potential concerns and in the safety or security aspects of our current research. So I guess I would say I'm generally for the thinking about how to harmonize at the international level, but I would want that to be focused not just on like the framework itself, but also harmonizing on, well, how are we learning from the implementation of this framework that, well, it doesn't work here, and it does work there, and it needs to be changed in this way. How do we have that iterative process itself built into the design of of uh, of the governance system? Yeah.
So next question. Oh, that's actually interesting. Could you please define what exactly is governance? Does it always come from the inside the community, or can it be external influence too?
That's interesting if that's the way that you heard my talk. Because that probably I was probably trying to push the internal drive more. So I don't know that there is a good like dictionary definition of governance, and I think if there is, I don't know if I would like it, but I would say that a useful way that I think about governance is if you think of the work that you do on a daily basis, there are all kinds of reasons why you do that work and not other work, and you do it in the way that you do and not in another way. All of those reasons govern what your work is. And so governance here, I take as a very broad brush term to attend to the contexts within which research is done, and who has a say, what systems are in place to focus our attention in some directions and not in others. That is so a lot of that comes from inside. Like, you know, if we go around the room here and talk about everybody's labs, and like, what is your, you know, what's your if we take a common lab routine that everybody probably does, I bet we would find like 10 different ways that people do that routine in this room, and that those different ways might lead to different processes for, you know, conducting your research, that might lead you to think in different ways about the research that you do and the ways that it might be good or not. So that's the inside, but it can also be external. So like all of these high level policies that I'm talking about, like dual use research of concern, things like that, the institutional biosafety framework, the, you know, human subjects research framework. These are top level policies that then get trickled down to institutions that then have to implement them. And so that kind of large external governance system itself has its own set of concerns around how to design it well, how to design it reflexively, and how to design it in a way that it can iterate. The dual use research of concern framework is supposed to be iterated upon based on the policy as written, and yet there hasn't been the systematic design to know how to iterate upon it on a, you know, on what kind of time scale.
I have my own question, but there's another one in the chat I see.
That's all right.
So the question is, oh, okay. So maybe I'll ask my question first.
Yeah.
I basically have a question. Do you have a weird science framework? Like, is there any way to look at, for example, to run a mouse analysis on something that, like in our field where we're using, let's say, a cell-free chassis to create a replication competent virus from a translation system that's orthogonal to natural translation system? Is there a standard framework to look at containment, kill switches for that? Or is that something that we should be working on because that haven't been done before?
Well, I'm not quite sure I understand the question. If you're asking me the question, it sounds like the answer is no. I'm not an expert in the cell-free system kind of world, right? So if that's the case, that you all have not yet have a conversation about how being cell-free and orthogonal might change the might require a change in the assumptions that we're making within our governance processes, then that is very much a conversation that needs to be had now. and
So
we need to figure out how to build that conversation up.
we kind of do, on and off. We do have those conversations in smaller groups. The Build a Cell community has safety and security working group. And as you know, I try to sneak in those topics into our EBRC safety working group, but I'm not getting much traction because people think this is too early to talk about it. And I don't think it's too early. I think this is just the right time when we're building those technologies.
One of the things that I often respond to in the "it's too early" kind of comeback is that there are lots of different ways to attend to the broader aspects of your research, depending on the stage that you're at. At these very early stages, one thing that's often happening is the creation of norms and standards for conducting research, creation of standard like organism models, the creation of like what counts as a good question in this research culture. And those themselves are really important moments of institutionalizing particular ways of looking at the relationship between the biology and the social structure of science and the broader structure of society and the problems that you think that this research is going to be good to address. And so attending to like really working with somebody like me, a social scientist and a science and technology studies person whose job it is to unpack the assumptions that are being made as science evolves, can be really productive in that moment, because once standards are set, like if we go back to Asilomar, like one of the outcomes of Asilomar was creating E. coli K12, which then becomes this model organism throughout the whole life science community. Like, why was it E. coli and not yeast? Like, there was a conversation that went on there. What other kinds of organisms could have been considered instead? Why was it so quickly narrowed down? And was that a good idea? Like, the same can happen in this space right now, and should happen, I would argue, because by bringing in those broader perspectives, you get a much richer attention to and purposeful attention to the ways that your research might be good and bad for the world.
That does make a lot of sense. I mean, we are doing it from the inside of the community, but we're also we need people from the outside. I see several people are signing off already because we're running up to the hour. So I just want to open the floor if anyone has any last questions on chat or in person. And I want to say thank you so much, Sam.
It's a delight being here.
And I'm sure that's going to catalyze a lot of conversations within our own.
As I showed on the slide, my email is samuel_evans@harvard.edu. Happy to continue the conversation in whatever way people would like.
Thank you so much.
Okay.
Thanks a lot. And I see a whole bunch of chat thank you messages. So thanks, everyone.
Great. Okay. Thanks, everybody.
Thank you. Bye.